A very good afternoon to everyone. Welcome in today's class. Today we are going to talk about gluconeogenesis. As we say in the beginning that let's crack NEET PG together. So the topic of today is gluconeogenesis. We are going to talk about gluconeogenesis. But before beginning with today's topic, let us have a small introduction about Unacademy. Unacademy is one of the India's largest platform for the preparation of NEET PG. India's best educators teach at Unacademy, so you can get this benefit of being mentored by the best faculty members all over India. Then there are special classes, and in these special classes, you can ask your doubts and queries directly with your faculty member. They'll be very much willing to solve your doubts and they'll be very much helping you out in clearing any type of confusion. Then there are test series, and as I always say, for qualifying any kind of examination, it is very much necessary that you should know the pattern of the examination, the PYQs. That is also possible with Unacademy with the help of test series. Then you can just go on Play Store and write Unacademy Learning app. You will see the logo of Unacademy. You can just download the app and make your account, and you are good to go. So what is the benefit of Unacademy is flexibility of schedule, language can be of your choice, and unlimited practice. There are many faculty members who are teaching in regional languages, so you can get this benefit of using your own language of choice. Then it is no different than a normal offline kind of teaching. As you can chat with your educator, there are interactive calls, then you can learn anytime, anywhere with the help of Unacademy. So this is the benefit of being a student of Unacademy, right? Then moving forward, there is this plus subscription, and what is the benefit of this plus subscription is that you can get all the 19 subjects at this plus subscription, and all the upcoming exams and all the next development in the NEET PG, you will be very much intimated with the help of plus subscription. Then there are short-term courses and crash courses for all the upcoming exams that can also be possible with the plus subscription. So this plus subscription, you need to pay a certain amount for this plus subscription, and that is quite pocket friendly, but still you can get a discount of 10% by using my referral code, that is FB01. So by using my referral code FB01, you will be getting an 10% discount in the subscription fees, which is quite nominal. And what I would suggest is go for a yearly or a 2 yearly program so that you can make the most out of it. So this is about Unacademy. Now moving towards the topic of the day, that is gluconeogenesis. Gluconeogenesis. This is the topic. So whenever there is formation of glucose from the non-carbohydrate source, it is called as gluconeogenesis. So it is formation of, first of all, just seeing the literal definition of gluconeogenesis. So that is formation of glucose from non-carbohydrate source, from non-carbohydrate source or precursor, is called as gluconeogenesis. So the non-carbohydrate source which we are writing over here, so what are these non-carbohydrate sources or precursors? Let us just see the names of the precursors which are being used for formation of glucose. So let me take pen. Yeah. So the non-carbohydrate source are lactate, then glycerol, then Propionyl-CoA, or odd chain fatty acid, and the glucogenic amino acid. The glucogenic amino acids. So from these four sources, whenever there is formation of glucose, it is called as gluconeogenesis. So don't forget lactate, glycerol, propionate, propionyl-CoA or chain fatty acids and glucogenic amino acids. Right? So we are going to talk about gluconeogenesis under following headings in today's class. That is, we are going to talk about the definition, then site of gluconeogenesis, then some of the important points regarding gluconeogenesis, and we are going to see the four substrates which we have just seen. Those four substrates, how are they used for gluconeogenesis? So four substrates we are going to talk about in detail, and then the amount of ATP which is utilized to form one glucose molecule, and then lastly regulation. So we are going to see gluconeogenesis under the following headings. So starting from definition that we have seen already, that is formation of glucose from non-carbohydrate precursors is called as gluconeogenesis, and that is from lactate, glycerol, propionate, propionyl-CoA or chain fatty acids and glucogenic amino acids. This we have seen. Now moving a little ahead in the topic, that is talking about some of the important things which are related to gluconeogenesis. So important things which are related to gluconeogenesis, important points. So whenever there is breakdown of glucose, it is called as glycolysis. But when we are talking about gluconeogenesis, it is not complete reversal of glycolysis. So it's complete, not complete reversal of glycolysis. First thing, why it is not complete reversal of glycolysis? So if you can remember, glycolysis has got three reactions which are irreversible in nature. So that's why the whole glycolysis cannot be reversed. So it is not a complete reversal of glycolysis. Then second thing, the three reversible steps which are there in glycolysis, they are reversed by four important enzymes. So three irreversible reactions of glycolysis are reversed by four enzymes. Then next point regarding reversal of glycolysis or gluconeogenesis is for production of one glucose. For production of one glucose, there is expenditure of 6 ATP. 6 ATP needed. Then it's a source of glucose. The next thing which is related to gluconeogenesis, it's a source of glucose. Source of glucose. Then when is this source of glucose actually needed? Because already when we are eating food, we are getting glucose from there. So whenever there is starvation or whenever there is this, after the meal condition, after the depletion of glycogen, after depletion of glycogen, after depletion of glycogen. So when does this glycogen stores they deplete? That is after 16 to 18 hours of meal. Gluconeogenesis will occur for production of glucose. Then lastly, this glucose is very important source of energy for brain and RBC. So important source of energy for RBC and brain. So these are the important points which are related to gluconeogenesis. Let us move a little ahead. That is the non-carbohydrate precursors which are used for the purpose. What are they? So let us take another slide. So the four substrates. The four substrates. We have already talked about these four substrates and we have just seen the name. So the first substrate we are going to see is lactate. Then we are going to see glycerol. Then we are going to see propionyl-CoA. Then we are going to see glucogenic amino acid. Glucogenic amino acids. So these are the four non-carbohydrate based substrates. So when we are talking about glucogenic amino acids, so amino acids, they fall into two categories: glucogenic amino acids and ketogenic amino acids, and some amino acids, they fall into the category of both glucogenic as well as ketogenic. So when we are saying glucogenic amino acids, so all the amino acids, they are somehow glucogenic except leucine. So just remember this thing, only leucine is exclusively ketogenic. Rest all are glucogenic, right? So this was about the four substrate, and when we are saying glucogenic amino acids, just the most important amino acid that you need to skip out is leucine, right? Let us move further. So what is the site of gluconeogenesis? Whenever we are talking about any kind of pathway, we always make sure to know that the place where this pathway is occurring. So what is the site of gluconeogenesis? So it is cytosol and mitochondria. It is cytosol plus mitochondria. So the important tissue in which glycolysis is going to, gluconeogenesis is going to occur is liver and to some extent in renal cortex. So these are the main tissues of the gluconeogenesis. Main tissue for gluconeogenesis, right? So all the steps are similar to glycolysis except three irreversible steps, and these three irreversible steps we need to reverse, and these three steps are going to be reversed with the help of four enzymes, right? So four enzymes for three irreversible steps. Fine. So the first thing that we are going to see is synthesis of glucose from pyruvate. Synthesis of glucose from pyruvate. The very first thing we are going to see this. So let us see this thing, that how the glycolysis occurs. So first of all, we draw this glycolysis so that we can always have this key in our mind, that which of these steps are irreversible and which of these steps are reversible. So from glucose, there will be formation of glucose. I need to make it a little compact. So those who are seeing this video live, they can write in the chat box whether they are able to understand or not. Then glucose 6-phosphate. Then from glucose 6-phosphate, there will be formation of fructose 6-phosphate. From fructose 6-phosphate, there will be formation of fructose 1,6-bisphosphate. Then from fructose 1,6-bisphosphate, there will be formation of two things, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And this reaction is reversible, and this reaction is reversible as well. This reaction is reversible. Then there will be formation of From glucose 3 for glyceraldehyde 3 phosphate, there will be formation of 1,3 biphosphoglyceraldehyde, and 1,3 PPG will change into 3 PPG, which will change into phosphoenol pyruvate, and this will change into pyruvate. So these reactions are reversible. So the reactions which are non-reversible, let us just highlight it with some other color. So this reaction is irreversible, this reaction is irreversible, and this reaction is irreversible. This is the reaction irreversible, this is the reaction irreversible. Now whole thing will be rotating around this thing, right? So how the pyruvate is getting converted into glucose, let us move towards that. So we have got this phosphoenol pyruvate which is getting converted into pyruvate, right? So let us start our story from pyruvate. So this pyruvate will be changing into oxaloacetate. And when pyruvate is getting converted into oxaloacetate, it is a kind of carboxylation reaction. So whenever a reaction is a carboxylation reaction, what three things are needed? ATP is needed, biotin is needed, and carbon dioxide is needed. So all these three things are needed for this reaction. And the enzyme which is doing this reaction is pyruvate carboxylase. It's pyruvate carboxylase. This oxaloacetate will now change into phosphoenol pyruvate. This is going to change into phosphoenol pyruvate. And the enzyme which is responsible for this reaction is phosphoenol pyruvate carboxykinase. Phosphoenol pyruvate carboxykinase. And this phosphoenol pyruvate, which in N changes into pyruvate. So this irreversible step, formation of phosphoenol pyruvate into pyruvate, is actually reversed over here. Let me take the laser point. So here if you can see, the formation of phosphoenol pyruvate into pyruvate is a non-reversible reaction. But how the phosphoenol pyruvate is formed from pyruvate by the formation of oxaloacetate by two steps. In the first step, there is use of pyruvate carboxylase, and in the second step, there is use of phosphoenol carboxykinase, right? So this is how phosphoenol pyruvate is formed from pyruvate. So this is the irreversible step in glycolysis, and here it is reversed by two enzymes. Now, the two enzymes is pyruvate carboxylase, which is actually ATP, biotin, and carbon dioxide dependent. Now, the reaction where the formation of phosphoenol pyruvate is occurring from oxaloacetate, again there will be use of GTP and there will be release of carbon dioxide. So in this step again there is expenditure of 1 GTP, here there is expenditure of 1 ATP, and the carbon dioxide which was utilized in the first reaction when pyruvate was changing into oxaloacetate, there was addition of 1 carbon dioxide. Now when oxaloacetate is changing into phosphoenol pyruvate, there is removal of 1 carbon dioxide, but for both of these steps there has been expenditure of 1 ATP. Right? So what is happening? The oxaloacetate is getting converted into phosphoenol pyruvate. there is one another fate of oxaloacetate, that is oxaloacetate gets converted into malate, which is transported out of mitochondria, and this malate is converted into oxaloacetate, and now in cytosol the oxaloacetate gives phosphoenol pyruvate. So what I am trying to say is this oxaloacetate does not get converted into phosphoenol pyruvate, but this oxaloacetate gets converted into, first of all, it gets converted into malate. So why is it getting converted into malate? Because when it is converted into malate, it can be transported out of mitochondria. So here we can say transported out of mitochondria. When it is transported out in the mitochondria, and when it is in cytosol, when when it is in cytosol, this malate is converted into oxaloacetate. When it is in cytosol, it is converted into oxaloacetate. And this oxaloacetate in cytosol, right? Here it was in mitochondria. So in cytosol, this oxaloacetate then gets converted into phosphoenol pyruvate. So this synthesis of glucose from pyruvate is not a two-step process, but because oxaloacetate cannot be as it is transported out into mitochondria, it needs to get converted into malate, and then malate is converted into oxaloacetate, and then in cytosol it is converted into phosphoenol pyruvate. So this step is again energy dependent and carbon dioxide is removed. So I hope you have understood that how we have done the formation of glucose from pyruvate. Right now, talking about the other reactions which are irreversible. So what are those irreversible reactions? Those we have seen. Let us go back to our glycolysis. So here, can you see? Let me take the laser. So can you see this fructose 1,6 bisphosphate cannot be converted into fructose 6-phosphate. So there is an enzyme which is responsible for the reversibility of this reaction. We will be talking about that. Let me take the pen and another slide. So this fructose 1,6 bisphosphate can get converted into fructose 6-phosphate, which is by nature an irreversible reaction, will get converted into a reversible reaction by the help of another enzyme, that is fructose 1,6 bisphosphatase. Bisphosphatase. This is the enzyme needed over here. So don't forget the name of the enzyme, that is fructose 1,6 bisphosphatase. Then there will be removal of one phosphate from fructose 1,6 bisphosphate. Right now, the another reaction which is irreversible, so that is glucose 6-phosphate cannot get converted into glucose directly by the help of hexokinase or glucokinase, because that is the enzyme which is responsible for formation of glucose 6-phosphate from glucose. So the same enzyme won't be reversing the step. So the enzyme which can reverse this reaction is glucose 6-phosphatase. Glucose 6-phosphatase. So remember the name of the enzyme. So it is not that difficult also, because in both of the cases above and here, both of the places you can see there is ase, phosphatase. That means there will be removal of one phosphate. So as easy as this, just remember phosphatase and there will be removal of phosphate. So talking about these enzymes, so glucose 6-phosphatase, specifically about glucose 6-phosphatase. Remember, this is very important for all the kind of MCQs. This enzyme is actually absent in muscle. Absent in muscles and adipose tissue. Absent in muscles and adipose tissue. So it is present only in liver and kidney. Liver and kidney. So there is no formation of glucose from glucose 6 phosphate in the muscles and adipose tissue, and this type of reaction occurs in liver and kidney, right? Thus free glucose is produced in liver and kidney. So in liver and kidney, free glucose is produced. Free glucose produced, right? And this is the common pathway for synthesis of glucose from non-carbohydrate precursors. So whenever we are talking about lactate or we are talking about propionyl-CoA, these will be the basics. This is how the basic pathway will occur. So this is the common pathway for generation of glucose from the non-carbohydrate precursors, right? So this we have seen. Now specifically talking about the precursors. So the first precursor that we need to see through which the glucose is formed is lactate. So the heading should be synthesis of glucose, synthesis of glucose from lactate. Synthesis of glucose from lactate. So how does this occur? This lactate will get converted into pyruvate, and we have seen how the pyruvate is getting converted into oxaloacetate and how it is converted into phosphoenol pyruvate. So oxaloacetate and oxaloacetate is getting converted into phosphoenol pyruvate. So we know from here pyruvate to phosphoenol pyruvate. We know this reaction. Now we need to see how the lactate is getting converted into pyruvate, and this is a kind of reversible reaction. The enzyme which is needed over here is lactate dehydrogenase enzyme, and this enzyme is actually NAD dependent. So there will be this is NADH dependent reaction. So the lactate will get converted into pyruvate with the help of lactate dehydrogenase enzyme, right? So this is how lactate gives rise to glucose. Now all the previous reactions will be same, like how the pyruvate is getting converted into phosphoenol pyruvate. We have seen that it's a basically two-step process, how malate is formed and how it is transported out of mitochondria and how it is giving rise to oxaloacetate, then phosphoenol pyruvate, and how fructose 1,6 bisphosphate. Let us go to the yes, how this fructose 1,6 bisphosphate is getting converted into fructose 6-phosphate with the help of enzyme fructose 1,6 bisphosphatase, and how this glucose 6-phosphate is getting converted into glucose, right? Now moving ahead, that is the next thing, that how the synthesis of glucose occurs from propionyl-CoA. So the next heading should be synthesis of glucose from propionyl-CoA. Propionyl-CoA. How does this happen? So this propionyl-CoA, specifically talking about propionyl-CoA. So this propionyl-CoA is actually a odd chain fatty acid. Odd chain fatty acid, just I'll write here, odd chain fatty acid, right? This is a odd chain fatty acid. Now, whenever there will be starvation, the first thing that will give in fatty acid, the first thing that will give rise to glucose is odd chain fatty acid. So during starvation, odd chain fatty acid gives glucose. Let us write over here, during starvation, odd chain fatty acid gives glucose. Now, how there is formation of phosphoenol pyruvate from propionyl-CoA. This we need to see. So, what are the enzymes which are required and what are the steps? So, first of all, we will write Propionyl-CoA because this is the precursor. So, Propionyl-CoA. Propionyl-CoA will get converted into methyl malonyl-CoA. Methyl malonyl-CoA. And this reaction is catalyzed by propionyl-CoA carboxylase. Propionyl-CoA carboxylase. So if you can see carboxylase, it means it's a carboxylation type of reaction. And carboxylation reaction, there will be use of A, B, C. A is for ATP, B is for biotin, and C is for carbon dioxide. So these things are needed in the first reaction for the formation of glucose from propionyl-CoA. Now the second step, the formation of malonyl-CoA will get converted into succinyl-CoA, and the succinyl-CoA will get converted into oxaloacetate, and oxaloacetate will get converted into phosphoenol pyruvate. Now the enzymes which are needed. So this methyl malonyl-CoA will be converted into succinyl-CoA by methyl malonyl-CoA mutase. This is the enzyme, and it is vitamin B12 dependent reaction. So the succinyl-CoA will be formed from methyl malonyl-CoA, and this succinyl-CoA is itself intermediate of TCA cycle. Then it is needed for heme synthesis. So various functions are there, and from succinyl-CoA there will be formation of oxaloacetate, and oxaloacetate will form phosphoenol pyruvate. So how propionyl-CoA is used up for the production of glucose that we have seen. So this basically occurs in starvation. So whenever there will be starvation, the odd chain fatty acid will participate in formation of glucose. So we have seen the two enzymes, propionyl-CoA carboxylase and methyl malonyl-CoA mutase, which is vitamin B12 dependent reaction for the production of phosphoenol pyruvate. Now moving a little further, that is synthesis of glucose from glycerol. So the next thing is synthesis of glucose from glycerol. Synthesis of glucose from glycerol. Right? So how will it happen? So first of all, the precursor is glycerol. So let us write glycerol. This glycerol will get converted into glycerol 3-phosphate, and this glycerol 3-phosphate will get converted into dihydroxy acetone phosphate. So the two enzymes which are required over here. So let us highlight the name of those enzymes. So glycerol is getting converted into glycerol 3-phosphate with the help of glycerol kinase. And glycerol 3-phosphate will get converted into dihydroxy acetone phosphate with the help of glycerol 3-phosphate dehydrogenase. Glycerol 3-phosphate dehydrogenase. So this is how the synthesis of glucose occurs from glycerol. So the two enzymes you need to remember is glycerol kinase and glycerol 3-phosphate dehydrogenase. So this was the third substrate in the series of production of glucose. The second substrate was Propionyl-CoA, and the first substrate was lactate. So we have seen three substrates for the production of glucose. Let us move further and let us talk about the fourth substrate. So our fourth substrate is glucogenic amino acids. So except leucine, we are going to talk about all the amino acids. So synthesis of glucose from glucogenic amino acid. Glucogenic amino acids. So except leucine. Now exclusively which amino acid participate over here? So the most common amino acid which participate in being a glucogenic amino acid. So you have to remember alanine. So alanine is the most common amino acid which is used for the production of amino acid. So alanine is mostly used for glucose production, right? So the amino acids which give intermediates of TCA cycle, let us talk about them. So first of all, pyruvate is given by alanine, it is given by serine, it is given by glycine, and it is given by cysteine. Pyruvate is given by these amino acids. Then second, succinyl-CoA. Succinyl-CoA. It is given by valine, methionine, isoleucine, and threonine. Succinyl-CoA is given by these four amino acids. Now third, alpha-ketoglutarate. Alpha-ketoglutarate. So alpha-ketoglutarate is given by glutamate, then glutamine, then proline, then histidine. So alpha-ketoglutarate is given by these four amino acids: glutamate, glutamine, proline, and histidine. Now the fifth intermediate, that is fumarate. So fumarate is given by tyrosine and phenylalanine. Tyrosine and phenylalanine. Right? Then the next intermediate, so I think I've done some mistake in the numbers here. So this fumarate is the fourth. Fumarate is given by tyrosine and phenylalanine, and the fifth one, that is oxaloacetate. Oxaloacetate. So oxaloacetate is given by aspartate, and it is given by asparagine. Aspartate and asparagine. So we have seen how there is production of the intermediates of TCA cycle through the amino acids. Now talking about the regulation, that how all these things are regulated. So the next thing that we are going to see now is regulation. So the four enzymes are the key regulatory enzymes of gluconeogenesis. So there are four regulatory or key enzymes. For regulation, there are 4 regulatory or key enzymes. So first of all, just let us highlight the name of those enzymes. That what are these enzymes which are necessary. So the enzymes are pyruvate carboxylase, pyruvate carboxylase 1. Then second is phosphoenol pyruvate carboxykinase, carboxykinase. The third one is fructose 1,6 biphosphatase, fructose 1,6 bisphosphatase. Then fourth enzyme is glucose 6-phosphatase. This glucose 6-phosphatase. So these are the 4 key regulatory enzymes for the reaction, 4 regulatory or key enzymes for the regulation of gluconeogenesis. So it is for pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase and glucose 6-phosphatase. So the enzymes, they are regulated actually by 3 ways. These 4 enzymes are regulated in 3 ways. Regulated by 3 ways. So let us talk about these ways through which they are regulated. So the first way through which they are regulated is induction and repression. Induction and repression. Induction and repression. So pyruvate carboxylase, pyruvate carboxylase pyruvate carboxylase, it is repressed by insulin. Repressed by insulin. It is repressed by insulin. But insulin, it favors glycolysis and inhibits gluconeogenesis. So insulin favors glycolysis but inhibits gluconeogenesis. Right? Then talking about the second enzyme, that is glucagon. Glucagon. So the second hormone, glucagon, it is responsible for the gluconeogenesis enzymes. What does it do? It induces the enzymes which are responsible for gluconeogenesis. So not alone glucagon, but glucocorticoids, they are also responsible for induces the gluconeogenic enzymes. Gluconeogenic enzymes. So insulin, it inhibits gluconeogenesis. Glucagon, it induces the gluconeogenic enzyme. Thus, it induces the gluconeogenesis. So this was the first way how the 4 enzymes, they are regulated, that is induction and repression. So repression of gluconeogenesis is by insulin and induction of gluconeogenesis is by glucagon. So here you can just write in a very small letter that this is the inducer of gluconeogenesis and that this is the repressor of gluconeogenesis, right? let us move further towards the second way by which the enzymes they are regulated. So the second way through which these enzymes they are regulated is through covalent modification. Covalent modification. Covalent modifications of enzymes. So how the covalent modification will actually induce the enzyme or repress the enzyme. So the way through which the covalent modification occurs is actually by the help of phosphorylation. So the key thing which happens in covalent modifications is phosphorylation. Phosphorylation reaction. Now, what happens is the cyclic AMP, whenever it is in high amount, so what will happen that it will induce and it will increase the amount of cyclic AMP dependent protein kinases. Dependent protein kinases. It will increase the amount of cyclic AMP dependent protein kinases and then phosphorylates the key enzyme. So these cyclic AMP dependent protein kinases, what thing actually? Cyclic AMP dependent protein kinases, now what are they going to do? They are going to phosphorylate the key enzymes as they are kinases. So phosphorylates the key enzymes in gluconeogenesis and activates the gluconeogenesis in gluconeogenesis and activates the pathway. The pathway. So I hope you are getting that how the covalent modifications are doing the thing. So first, whenever there will be increased amount of cyclic AMP, it will in turn increase the concentration of cyclic AMP dependent protein kinases, and the cyclic AMP dependent protein kinases phosphorylates the key enzyme in gluconeogenesis and activates the pathway. So this was about the covalent modification of the enzymes which are responsible for the regulation of the four enzymes. First one is pyruvate carboxylase, second is phosphoenol carboxykinase, third is fructose 1,6 bisphosphatase, and fourth is glucose 6-phosphatase. Right? Let us move further and talking about the third way through which the things are regulated. So that is the allosteric regulation. That is allosteric regulation. Allosteric regulation. So the two important things which we need to know in allosteric regulation, two important things. What are those? Acetyl-CoA. First is acetyl-CoA. Acetyl-CoA. What is this acetyl-CoA? So this is the allosteric activator of pyruvate carboxylase. So this is the allosteric activator of pyruvate carboxylase. Acetyl-CoA is the allosteric activator of pyruvate carboxylase. Second that you need to know is fructose 2,6 bisphosphate. Fructose 1,6 bisphosphate. It is a negative activator or inhibitor of pyruvate carboxylase. So better we will write inhibitor of pyruvate carboxylase. It is the inhibitor of pyruvate carboxylase. So this is how totally the enzymes they are regulated, and how various non-carbohydrate precursors are used for the production of glucose. And in production of one glucose molecule from the non-carb sources, there is expenditure of 6 ATP needed, right? So before ending today's session, let us have a very quick revision of the things that we have seen in today's class. So we were talking about gluconeogenesis, that is the production of glucose from non-carbohydrate precursor. So the definition of gluconeogenesis is formation of glucose from non-carbohydrate precursors, specifically lactate, glycerol, propionate, Propionyl-CoA or odd chain fatty acid, and glucogenic amino acids, right? Then we saw this whole thing under the headings of definition, site where it is done, then important points, then the four substrates which are used for the production of glucose, then the ATP which is utilized in this production of one glucose molecule. Then we have seen the regulation. So have a quick revision. So the important points which are related to this is, it is not the complete reversal of glycolysis. This is very important to remember, that it is not just a complete reversal of glycolysis, because there are three steps which are non-reversible in glycolysis. So right, so it is not a complete reversal of glycolysis. So the three reversible steps of glycolysis are actually reversed by four enzymes. Over here, for production of one glucose, there is expenditure of 6 ATP. Then the sources of glucose, which is used like this through gluconeogenesis, so that occurs after 16 to 18 hours of meals, wherever there will be depletion of glycogen in the body. And this glucose is important for the proper functioning of RBC and brain. Then the four substrates is lactate, glycerol, propionyl-CoA, and glucogenic amino acids, and only leucine is exclusively ketogenic. So all the amino acids. Now the site where this occurs is in the cytosol and mitochondria, and the main tissues are liver and to some extent renal cortex. So the four enzymes, they are used for the reversing of the three irreversible steps. Now the synthesis of glucose from pyruvate. The first thing, how the glucose is formed from pyruvate. So this pyruvate gets converted into oxaloacetate with the help of a carboxylation reaction catalyzed by the enzyme pyruvate carboxylase. This oxaloacetate is formed in the mitochondria. It cannot be transported as it is. So it is converted into malate and it is transported into mitochondria, and malate is converted into oxaloacetate into cytosol, and this oxaloacetate, with the help of carboxykinase, which is an energy dependent step, and there is removal of carbon dioxide, gets converted into phosphoenol pyruvate, and the carboxylation reaction will go as it is. Right, so this is how there is formation of phosphoenol pyruvate from pyruvate, and rest of the two steps which are not reversible, that also we are going to see. So before that, let us have a look at which are the steps which are actually irreversible. So formation of glucose 6-phosphate from glucose is irreversible, and formation of fructose 1,6-bisphosphate from fructose 6-phosphate is irreversible, and formation of pyruvate from phosphoenol pyruvate is irreversible reaction. So this is our glycolysis, and these steps are irreversible, and these steps we need to reverse for production of glucose. So we have seen how pyruvate is changed into phosphoenol pyruvate, and fructose 1,6-phosphate gets converted into fructose 6-phosphate by the help of enzyme fructose 1,6-bisphosphatase, and glucose 6-phosphate is converted into glucose with the help of glucose 6-phosphatase. But the twist in glucose 6-phosphatase is that it is absent in muscle and adipose tissue, and it is present in liver and kidney. So the free glucose are mostly produced in liver and kidney. The free glucose is produced in liver and kidney. Then synthesis of glucose from lactate. So lactate gets converted into pyruvate by lactate dehydrogenase enzyme, and we know the further story how pyruvate changes. Then synthesis of glucose from propionyl-CoA. So this occurs during the starvation. The odd chain fatty acids will be utilized. So propionyl-CoA will get converted into methyl malonyl-CoA, which get converted into succinyl-CoA, and further succinyl-CoA will get converted into oxaloacetate and phosphoenol pyruvate. So the enzymes needed over here is propionyl-CoA carboxylase. First reaction is the carboxylation reaction. The second step requires methyl malonyl-CoA mutase, and it is a vitamin B12 dependent step. Then formation of glucose from glycerol. So there will be the utilization of enzyme glycerol kinase, and it will form glycerol 3-phosphate, and it will lead into formation of dihydroxyacetone phosphate, which is the intermediate. And rest of the reactions, they are reversible, and the place where the irreversible reactions are reached, we will just use fructose 1,6-bisphosphatase and glucose 6-phosphatase. Now synthesis of glucose from glucogenic amino acids. So mostly alanine is used for this purpose, and we are seeing that which amino acid is giving which precursor. So alanine, serine, and glycine, they give pyruvate. Then valine, methionine, isoleucine, and threonine, they give succinyl-CoA. Glutamine, proline, and histidine, they give alpha-ketoglutarate. Then tyrosine and phenylalanine, they give fumarate, and aspartate and asparagine, they give oxaloacetate. Then how is it regulated? So it is regulated by 3 enzymes. So they are pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. So the three way they are regulated is by induction and repression. So insulin is the repressor of gluconeogenesis, and glucagon is inducer of gluconeogenesis. Then through covalent modification, phosphorylation is used in covalent modification, specifically phosphorylation will occur. So cyclic AMP, whenever it will increase, it will lead into production of high amount of cyclic AMP dependent protein kinase, and they will phosphorylate the key enzymes, and when there will be phosphorylation of the key enzymes of gluconeogenesis, it will lead into activation of the pathway. Then we have seen allosteric regulation. So acetyl-CoA, it is the activator, allosteric activator of pyruvate kinase, and fructose 1,6-bisphosphate is inhibitor of pyruvate carboxylase. So this is how we have completed gluconeogenesis in detail in today's class. So this is it about for the gluconeogenesis. Hope you have understood the whole concept well. So by this you will be able to write your university examination, as well as you will be capable of solving all the MCQs related to the topic. So just go through the video systematically and try to keep all the things in mind. It will be very helpful in writing your theory paper, as well as it will be very much helpful in solving all the MCQs. So lastly, please subscribe our channel, that is Let's Crack NEET PG, and also hit the bell icon for getting all the notifications of the upcoming videos on our channel, that is Let's Crack NEET PG. And lastly, please use my referral code, that is FB01, for getting 10% discount in the Plus subscription, which can be highly beneficial for cracking the examination. So use my referral code, that is FB01. Do not forget my referral code, that is FB01. And if you have gained something, you like my video, you have understood the concept, please give it a thumbs up. It really motivates us when we see your likes on our video. It really motivates us. So that is it for today's class. We'll be meeting in another class with some other important topic of biochemistry. And also do the thing, if you want to read some important topic or some topic of your choice in biochemistry, or which you are finding difficult, So you can just write in the chat box or in the comment box that you want to read that particular topic, and I'll be very much willing to solve your doubts and queries, and I'll try to make the videos of the topics of your choice. So this is it for today's class. We'll be meeting again tomorrow with some other interesting topic. Till then, be happy, keep on studying.